Hello everyone, my name is Romaista Jutsa from Morocco. I'm a PhD student at the University of Ahmed of Fes. My research topic is combining artificial intelligence with lab-based experiments to tackle antimicrobial resistance. So as a PhD student, I recognize the global threat of antimicrobial resistance and the important role that bacterial genomics play in understanding resistance mechanisms. One of the powerful tools I have learned during this workshop is the whole workflow of next-generation sequencing, from sample preparation and library construction to sequencing using lysine.
My name is Roland Peter Komzuta from Ghana. I work as a biomedical laboratory scientist with the Ghana Health Service, specifically with the Eastern Regional Hospital, Koforidua. I joined this program because I wanted to gain deeper insight into antimicrobial resistance, especially using genomic tools like an NGS to decipher resistance patterns in bacteria. So when I found the form, I applied, and here I am on the program.
In 2023, we submitted a request to GSK to partner with us to organize 2 training workshops for African scientists. One workshop was to build capacity in microscopy and bioimaging, and the second workshop to build capacity in genome sequencing and bioinformatics. We received a lot of applications all across the African continent. This showed the high level of interest of participants to build their capacity in the subject areas. Due to the particular interest in the genome sequencing and bioinformatics workshop, we then submitted a renewal request to GSK to train more participants in these subject areas. We received approval from GSK for this subsequent renewal, and we are currently in the process of organizing these workshops. We received over 1,800 applicants for the genome sequencing and bioinformatics workshops from 47 different African countries, and we had to select 20 participants for each category of workshops, one for the intermediate level and one for the beginners level.
My name is Abiola, and I'm a research fellow at WACCBIP. Equally, I'm one of the facilitators of the AMR genomic workshop funded by GSK. Antimicrobial resistance is a global crisis with an annual mortality rate of 1 million, which might increase to 10 million by 2050 if we do nothing about it. The key challenge with antimicrobial resistance, especially within Africa, is lack of expertise. To bridge this gap, GSK has provided an opportunity to train the next generation of AMR fighters. And for the past 2 weeks, we have been guiding and training young African researchers from different parts of Africa. This is to say that we are building relevant capacity that can help us to deal with the challenge of antimicrobial resistance.
And then to act. It will act rapidly and then deal with it. So that's the implementation.
Because we do have the GSK funding, we are able to pull together young people, young scientists from east to west, south, and do this basic training. That is in 2 parts, a wet lab, NGS training that allows people to learn the workflows from DNA extraction or nucleic acid extraction to library preparation and next generation sequencing towards the identification of antimicrobial resistance. The other group looks at the downstream analysis, the bioinformatic tools that will enable them to do AMR detection. So you take raw sequence data, you clean it, you do that rigorous QC, and then you do genome assembly, and you are able to build draft genomes that we can now use other related tools and databases like the CARD database, the Patrick database, to detect AMR resistance and specific pathogens that we have sequenced as part of this training.
The support that we receive from GSK has been extremely helpful. And this is because we have enough resources to be able to help even broaden the scope of the training and provide enough time for the training. So we end up doing a 2 weeks intensive course, and this allows us to give in-depth training to the participants, which is more helpful than having shorter courses because of limitation due to the limit of resources. The approach that we use, or the methodology that we use for training in our workshop, is more of a hands-on approach, and this is basically because of the nature of our work. So we want the participant to be able to have an experience and build confidence when they are working in the lab. So we allow the participants to practice on actual samples all the way from the beginning. You have the participants who work with actual samples and see the entire process from sample QC, library preparation, library QC, all the way until they load the sample into the machine and they're able to generate the data. And this is a method or an approach that we've seen is more impactful as opposed to just talking to the participants and explaining to them what they are supposed to do.
My name is Mary Kameni. I'm a research student at the Molecular Diagnostic Research Group at the University of Yaoundé 1 in Cameroon. So actually, I'm a third-year PhD student. I have been struggling with my research, with some parts of my research. I really wanted to learn more on next-generation sequencing, and I was opportune to be selected among the 20 participants for hands-on training in WACCBIP on antimicrobial resistance, genomics of bacterial and bioinformatics. So coming to WACCBIP in these 2 weeks has really enhanced my skills because I wanted to learn about next generation sequencing for so long. I've tried several opportunities, but I couldn't get it. So this opportunity enabled me to gain hands-on on whole genome sequencing of bacteria using the Illumina preparation kit, from library preparation, going through the DNA extraction, DNA quantification, going through the documentation, and all the steps for sequencing. So I'm happy and I'm thankful to WACCBIP, and I'm equally thankful to GSK for granting me this opportunity, because the invaluable resources I've gotten here, the skills I've learned here are going to really help me to pursue my research activities back home in Cameroon and to be able to complete with my research objectives till I will defend my PhD. So I'm very grateful for this opportunity. Thank you very much.
Hi, my name is Victor Mosha. I am a laboratory scientist from the Kilimanjaro Clinical Research Institute in Tanzania. My motivation to attend this workshop is based on my upcoming project on the surveillance of Neisseria gonorrhea in Tanzania. In this project, we tend to do both phenotypic and genotypic characterization of gonorrhea. And my institute is well equipped with both molecular and microbiological equipment, but we are underutilizing it. So I want to use the knowledge and skills gained from this training to effectively utilize the equipment that we have in my institute to do the genomic work and phenotypic work for the Neisseria gonorrhea. I'm very grateful for WACCBIP and GSK for organizing this training. I know it is very expensive organizing such kind of training, and it is not even easy to get opportunity to do this kind of training. So for me, getting this opportunity, I am really grateful.
My name is Namachao Mindu. I come from Togo. I'm especially in Institut National d'Hygiène of Lomé. I thank the director of WACCBIP, Professor Gordon, and all the rest of the team of WACCBIP for doing a lot of work to help, and in this project of training the young people in bioinformatics and AMR.
Hi, my name is Amina Saliu. I'm a veterinary epidemiologist. I'm from Nigeria.
I have been opportune to partake in the WACCBIP training on antimicrobial resistance, bacterial genomics, and bioinformatics. It's been very explicit, it's been very interesting. It's very wonderful because I've learned so much. As a researcher in antimicrobial resistance and food safety, I recognize the growing need of using bioinformatics in research, in discovering the resistance mechanism of the bacteria and also the evolution. I have been able to advance my computational skills in bioinformatics. I've learned so much. I've learned how to do bacteria assembly, annotations, use of BLAST, the antimicrobial resistance databases. I've learned so much. The facilitators have been wonderful. The colleagues, researchers, have been able to network with fellow like-minded researchers. I've also met experts. I've been taught by experts in the field, and it's been very impactful.
Teach others, even empower others. In fact, if you have the capacity, we are also organize a workshop for a few people. And transfer the skills. Amplify the effect. And tell them about WACCBIP. And invite them to also come here when they get the opportunity. So building a community of scientists for the future of Africa. There's a lot of talent on the continent. We believe that African scientists, we can compete with anybody in the world if we're given the right support and the right environment. And so we believe that once you come here and have this experience, you can go and try to also make a difference where you are coming from. And we'll continue to build a community and strengthen each other and empower African science. So we thank you for staying in touch with us. Come back again. And wish you great success wherever you go. Tell us about your success, because these things help us to get support for what we're doing.
We'd like to thank GSK for this support, and we hope to continue partnering with them to build capacity for African scientists on the continent. Thank you, GSK.